Bacterial cell wall, gram-positive, gram-negative. A comprehensive visual guide to bacterial structures, staining mechanisms, and clinical significance. Fundamental biology. Introduction to the bacterial cell wall. External structure. A rigid complex semi-permeable layer located immediately external to the cytoplasmic membrane. Think of this as the bacterium's exoskeleton, sitting right outside its delicate inner cell membrane. It's a crucial interface with the outside world. Primary functions. Provides characteristic shape like cocci or bacilli and robust structural support to the cell. This is why a staphylococcus is round and a bacillus is rod-shaped. Without this wall, bacteria would lose their form and integrity. Osmotic protection. Prevents osmotic lysis or bursting when bacteria are in hypotonic environments. Bacterial cells are full of dissolved substances. In watery environments, water rushes in. The strong cell wall resists the pressure like a sturdy balloon. Chemical composition. Mainly composed of peptidoglycan or murine, varying significantly between gram-positive and gram-negative bacteria. Peptidoglycan is the universal building block, but how much of it there is and what surrounds it creates the major divide we see in staining. The diagram shows the general bacterial anatomy, highlighting the cell wall and plasma membrane. You can see labels for components like N-acetylmuramic acid and peptide chains, illustrating the basic structure we'll explore next. Structural biology. Structure and function of the cell wall. Peptidoglycan matrix, a mesh-like polymer of NAG and NAM sugars forming long glycan chains, the structural backbone. Imagine a fabric woven from long sugar strands. This fabric, the glycan backbone, gives the wall its basic shape and flexibility. Cross-linking mechanism. Short peptide chains cross-link the sugar backbones created by transpeptidase enzymes known as PBPs. These peptide chains act like stitches, sewing the sugar strands together into a tight, net-like sac. The enzymes that do this stitching are prime targets for antibiotics. Mechanical strength. Provides immense tensile strength to withstand high internal turgor pressure up to 20 atmospheres. That internal pressure is enormous, comparable to the pressure in a car tire. The peptidoglycan mesh is strong enough to contain it. Osmotic barrier. The rigid mesh prevents cell rupture in hypotonic environments while allowing diffusion. It's selectively permeable, keeping the cell from bursting but still letting essential nutrients and waste pass through. The diagram, a peptidoglycan cross-section, clearly shows the alternating NAG and NAM sugars forming the glycan chains, with the peptide side chains and crosslinks creating the dense and supportive mesh. Now let's examine the first major architectural style. Structural biology, gram-positive cell wall structure. These bacteria have a simpler but formidable design. Thick peptidoglycan, 20 to 80 nanometer multi-layered mesh that retains crystal violet stain. This immense thickness is their defining feature. It's like wearing a very thick, sturdy coat that soaks up and traps the purple dye during staining. Teichoic acids. Contains wall teichoic acids and lipoteichoic acids, providing negative charge and aiding adhesion. These acids stud the peptidoglycan layer. Their negative charge helps with ion movement and allow the bacteria to stick to surfaces, which is often the first step in causing an infection. Single membrane. Possesses a single inner plasma membrane, lacks an outer membrane and periplasmic space. Their architecture is straightforward, a thick peptidoglycan coat draped directly over a single cell membrane. There's no second outer membrane. Common examples. Representative genera include Staphylococcus, Streptococcus, and Bacillus. These are common causes of infections, from strep throat and staph skin infections to anthrax. Their thick wall is a key part of their identity. The cross-section diagram contrasts a gram-positive envelope with others. It highlights the prominent thick peptidoglycan layer as the dominant structural element. Now let's examine the more complex architecture of the other major group. Cell architecture, gram-negative cell structure. These bacteria have a more sophisticated, multi-layered defense system. Thin peptidoglycan, a very thin layer, only 2 to 3 nanometer, comprising only 5 to 10% of the cell wall weight, located in the periplasm. Their peptidoglycan is just a thin single-layer liner. It provides some shape but is not the primary protective barrier. Outer membrane and LPS, protective outer bilayer containing lipopolysaccharides or endotoxin and porin channels for transport. This is their signature feature, a second outer membrane. The LPS or endotoxin on its surface can trigger severe immune reactions like septic shock. The porins are like selective gates. Periplasmic space, a large gel-like space between inner and outer membranes containing enzymes and transport proteins. This space is a busy cellular compartment. It contains proteins that break down nutrients and manage what enters and exits the cell proper. The diagram, a gram-negative envelope, shows all these layers clearly. The outer membrane with LPS, the thin peptidoglycan in the periplasmic space, and the inner plasma membrane. Comparative analysis. Key differences: gram-positive versus gram-negative. Let's put them side by side to solidify these critical distinctions. On the left, gram-positive with examples like Staphylococcus and Streptococcus. On the right, gram-negative with examples like E. coli and Salmonella. Peptidoglycan layer. For gram-positive, it's thick, 20 to 80 nanometers, a multi-layered mesh. For gram-negative, it's thin, 2 to 3 nanometers, a single layer in the periplasm. This is the most fundamental structural difference and the direct reason for how they stain. Gram staining. Gram-positive retains the purple dye crystal violet. Gram-negative appears pink or red from the saffron and counter stain. The thick peptidoglycan of gram-positives traps the purple dye complex. The thin layer of gram-negatives cannot hold it, so they take up the pink counter stain instead. Unique components. Gram-positive have teichoic acids and lipoteichoic acids. Gram-negative have lipopolysaccharides or LPS and porins. These unique molecules dictate additional properties: adhesion and charge for gram-positives, toxicity and selective permeability for gram-negatives. Membrane structure. Gram-positive have a single inner plasma membrane only. Gram-negative have dual membranes, inner and outer. This double membrane of gram-negatives creates a significant extra barrier against antibiotics and disinfectants. Laboratory technique. Gram staining mechanism. A differential staining technique that classifies bacteria into two groups based on their cell wall structure. This is one of the most important and enduring techniques in all of microbiology. It's a simple series of steps that reveals a world of difference. Step 1. Crystal violet, the primary stain. Basic dye dissociates into Cv+ and Cl- ions, penetrating both cell walls and staining all cells purple. Initially, every bacterial cell on the slide is stained a uniform deep purple color. The dye gets into both the types of walls. Step 2. Gram's iodine, the mordant, acts as a trapping agent by forming large insoluble crystal violet iodine complexes within the cell. The iodine doesn't add color, it acts like a fixative. It binds with the crystal violet to form a bigger complex that is harder to wash out. Step 3. Alcohol or acetone, the decolorizer, dissolves the outer membrane of gram-negative cells, washing out the dye. Gram-positive cells dehydrate, trapping the dye. This is the critical step. The alcohol strips away the outer membrane of gram-negatives, letting the purple complex escape. In gram-positives, the alcohol shrinks the thick peptidoglycan, trapping the complex inside. Step 4. Safranin, the counterstain, stains the decolorized gram-negative cells pink or red. Gram-positive cells remain purple. Finally, a second, lighter dye is applied. It stains the now colorless gram-negatives pink, providing a clear visual contrast. The gram-positives, still purple, don't take up this pink dye strongly. The diagrams for each step visually track the dye in purple and pink cells, showing exactly what happens to each type of bacterium at each stage. Medical microbiology. Clinical importance. Antibiotics.
Understanding cell wall structure isn't just academic, it's the foundation for many of our most important life-saving drugs. Selective toxicity target. Peptidoglycan synthesis is an ideal drug target as human cells lack cell walls, minimizing side effects. This is the concept of a magic bullet. We can attack a structure that bacteria absolutely need but that our own human cells don't even have. Beta-lactams mechanism. Penicillins bind to PBPs, inhibiting transpeptidation or cross-linking, leading to osmotic lysis. Drugs like penicillin jam the transpeptidase enzymes, the ones that stitch the wall together. The bacterium tries to grow but can't make a strong wall, so it bursts from osmotic pressure. Vancomycin action binds D-ala-D-ala terminals, physically blocking cross-linking, primarily effective against gram-positive. Vancomycin is a large molecule that physically clogs the building sites on the peptide chains. It's too big to get through the outer membrane of most gram-negatives. Gram-negative resistance. Outer membrane blocks large molecules, porins and efflux pumps reduce intracellular drug concentration. This is a major clinical challenge. The gram-negative outer membrane acts as a formidable gatekeeper, and the bacteria have pumps to eject any drug that does get in. Common examples: beta-lactams like penicillin and cephalosporins, glycopeptides like vancomycin and bacitracin. These are classes of antibiotics you've likely heard of. They all work, in different ways, to disrupt deconstruction or integrity of the bacterial cell wall. Conclusion, summary and key takeaways. Let's bring together everything we've learned about these remarkable microbial structures. Understanding bacterial architecture is fundamental to microbiology and medicine. The cell wall is more than just a shell. It defines the bacterial's interaction with the world and our strategies to combat it. Structural architecture. The fundamental difference lies in the peptidoglycan layer thickness and membrane organization. Gram-positive bacteria rely on a thick peptidoglycan shield, while gram-negative bacteria utilize a complex outer membrane barrier. One is a fortress with thick walls, and the other is a castle with a complex outer gate and a thin inner wall. Both are effective, but in different ways. Diagnostic value. Gram staining remains the gold standard for rapid identification. The structural differences dictate the color retention, guiding immediate clinical decisions. In a matter of minutes, this simple test tells a clinician a tremendous amount about the likely cause of an infection and what drugs might work first. Clinical implications. Cell wall synthesis is a prime target for antibiotics. The outer membrane of gram-negative bacteria creates a natural resistance barrier. This explains why some antibiotics only work on gram-positives and why gram-negative infections can be trickier to treat from the start. Future research. Developing agents that can permeate the gram-negative outer membrane or inhibit novel cell wall targets is critical for combating antimicrobial resistance. As bacteria evolve resistance to our current drugs, understanding their walls in even greater detail is key to designing the next generation of therapies. Thank you. I hope this journey through the architecture of bacterial cells has given you a clear picture of a world that, though invisible, has an immense impact on our lives. Remember, these fundamental structures are the reason behind some of our oldest diagnostic tests and our most vital medicines. Keep that curiosity alive as you continue to explore.